student, in this class I will discuss the DNA replication. What is the DNA replication? The DNA replication is nothing but the process by which DNA molecule make its identical copy. These are known as DNA replication. That means if a DNA can form its own copy, these are known as DNA replication. And this function of our DNA is known as autocatalytic function. Is known as autocatalytic function. But if the DNA can form the RNA and protein, this function of our DNA is known as heterocatalytic function. Is known as heterocatalytic function. The process by which DNA molecule make its identical copy, these are known as autocatalytic function. But if the DNA can form the RNA through the process of transcription, and the formation of a protein from RNA, the process is known as translation. That means, what is the transcription and translation? The transcription and the translation is nothing but, transcription means the formation of RNA from DNA is known as transcription, and translation means the formation of a protein from RNA. These are known as translation. Especially the protein are formed by only mRNA. Is clear? Transcription means the formation of RNA from DNA, and the RNA is three types. One is mRNA, another is RNA, another is tRNA. The M stands for messenger RNA, R stands for ribosome RNA, and T stands for transfer RNA. But formation of a protein from only mRNA, these are known as translation. Is clear? And this function of our DNA is known as heterocatalytic function. And the DNA replication, this process are mainly differentiated into three types. That means the three types of our DNA replication. One is conservative. Second one is dispositive. And the third one is semi-conservative. Out of the three DNA replication process, the conservative and the dispositive, there was no experimental proof. But in case of a semi-conservative DNA replication, there was having a experimental proof. That means present experimental proof. And these experimental proof are given by two scientists, and these scientists are Michelson and Stahel. Is clear? The DNA replication are three types. One is conservative, another is dispositive, another is semi-conservative. Out of the three DNA replication process, the first two process, that is conservative and dispositive, there was no experimental proof. But in case of last one, that is semi-conservative DNA replication, there was experimental proof which is present. And these experimental proof are given by or prove that the two scientists are Michelson and Stahel. Then I can discuss what is the process of a conservative, dispositive and semi-conservative. The first one is conservative. The first one is conservative. What is the conservative mode of a DNA replication? And I can told that this conservative DNA replication, there is no experimental proof, and this process does not affect. What is the conservative? This name suggests the both strands are conserved. Conservative means both the strands are conserved. That means, suppose these are the two parent strands. Suppose these are known as parent DNA. These are known as both strands of a parent DNA. In the process of a conservative, it is told that the two nuclear strands is formed. The two nuclear strands is formed. The two nuclear strands is formed. One has both parent strands. One has both parent strands. And other has both the new strands. Both the new strands. These are known as, these are two new strands. Is clear? The DNA replication is a three type. I can discuss the first type. The first type is conservative mode of DNA replication. And conservative mode DNA replication means this name suggests both the strands are conserved. That means if your parent strand can form the two daughter strand, out of the two daughter strand, the one daughter strand are similar to this parent strand, and another strand are a newly strand. That means completely different to the parent strand. These are known as conservative mode of DNA replication. And this replication process, there was no experimental proof. Is clear? Then second one is dispersive mode of DNA replication. Dispersive mode of DNA replication. And this replication process, there was no experimental proof. And this replication process told that if your parent strands, suppose these are the parent strands, this parent strand are break. These parent strand are break several points. This parent strand are break several points, and each piece, these are known as first piece, these are known as second piece, these are known as third piece, these are known as fourth piece. Similarly, 1, 2, 3, and 4. And each piece are a replicate to form new copy. Each piece are a replicate for new copy, and both the parent strand and the new strand combine to formation a mixture of the strand. Mixture. That means both the new strand and the parent strand are present in patchy appearance. That means the conservative dispersive DNA replication is told that the one parent strand is formed two daughter strands, and each daughter strand have both the parent strand and the new strand. This black line represent this parent strand, and this blue line represent the new strand. These are the new strands. Similarly, second one, parent strand patches and intermediate between two parent strands, there was a daughter strand is also be present in this manner. Is clear? The second replication process is a dispersive mode of a DNA replication. And this dispersive mode of a DNA replication is told that the two nuclear strands is formed from parent strand. These are known as parent strand. and these parent strand are break several points and each piece replicate and randomly join to formation this patchy appearance to new daughter DNA. Is this clear? That means this black color is represent the parent strand, and this blue color is represent the new strand. Is new strand. Similar in case lower case. Is clear? Then third one is semi-conservative mode of our DNA replication. What is the semi-conservative mode of our DNA replication? It is very, very important because there was experimental proof is present, and these experimental proof were given by two scientists. These scientists are Messerson and Staff. That means the third one is semi-conservative mode of DNA replication. We know that conservative means both the parent strand are conserved, but semi-conservative means this name suggests the semi means half. Semi means half. That means half of the parent strand is conserved. These are known as semi-conservative. That means suppose these are the parent strand. Suppose these are the parent strand. I can represent the blue line. These are known as two parent strands. And these two parent strands, this one parent strand, are divided or replication for two daughter strands, and each strand have a new strand and a parent strand. Each strand have a new strand and a parent strand. Each strand have suppose these are the parent strands and these are the new strands. Similarly, these are the parent strands and these are the new strands. That means this semi-conservative DNA replication told that the two new DNA molecule is formed and each DNA molecule have one parent strand and one new strand. Each daughter molecule have one parent strand and other is a new strand. These are known as semi-conservative. We clear that? First I will discuss the DNA replication is three types. One is conservative, another is dispersive, another is semi-conservative. Conservative means both the parent strand is conserved. Dispersive means the DNA breaks several points and each piece replicates and randomly join. And third one is most important. This semi-conservative DNA replication told that the half of parent strand is conserved. That means in this case, the two new DNA strand is formed and each new DNA strand, that means each daughter strand have a parent strand, ଗୋଟେ parent strand contain करथबो and a new strand ମଧ୍ୟ ଗୋଟେ contain करथबो. One new strand contain करथबो. These are known as semi-conservative mode of DNA replication.